Hello everybody, my name is Fabien van der Meers and I'm R&D Technical Leader and Quality Assistant. Today I'm going to present data on the characterization of anti-HCP antibodies by 2D differential gel electrophoresis and especially on the implementation of the Melanie Coverage software. The production of complex recombinant proteins, such as monoclonal antibodies, requires a system that has the cellular machinery capable of processing, folding, assembling, and post-translationally modifying the product to generate a required target protein. Today, 95% of the monoclonal antibodies are produced in mammalian cells, and especially on Chinese hamster ovary cells, or CHO. CHO are the best choice as a mammalian host for industrial production of recombinant proteins for different reasons. They grow fast and are ideal for large-scale cultures. The transfection rate is very efficient. They allow a high productivity, 1 to 10 grams per liter, and adequate post-translational modification can be made in CHO. During the culture, the recombinant antibody is secreted from the cell into the surrounding media. Host cell proteins, or HCPs, can be co-extracted with the antibody, and it is therefore necessary to purify the product using a series of downstream processing steps. These steps are designed to remove all the contaminants as host cell DNA, RNA, and proteins. As described in these pictures, they can include centrifugation, filtration, or chromatography steps. So, host cell proteins are process-related impurities derived from the host organism used for the production of monoclonal antibodies. Such residual HCPs carry substantial risk of decreasing efficacy of the biotherapeutics and causing adverse immunogenic reaction in patients. Therefore, HCPs are a safety and quality issue. Therefore, regulatory authorities request the detection of residual host cell proteins contaminants and the development of methods that reduce them to the lowest acceptable levels. For example, we can find here different extracts of ICH guidelines. ICH Q6b tells that process-related impurities may include cell culture media, host cell proteins, DNA. These impurities should be minimized by the use of appropriate, well-controlled manufacturing processes. For host cell proteins, a sensitive assay capable of detecting a wide range of protein impurities is generally utilized. In ICH Q7, where appropriate, the removal of media component, host cell proteins, other process-related impurities, product-related impurities, and contaminants should be demonstrated. Different techniques can be developed to monitor the presence of HCPs in the final product. As seen in this table, techniques can be divided in immune-specific methods and non-specific methods. Immune-specific techniques use specific antibodies to detect the HCP and include ELISA, thresholds, Western blot, dot blot, or biosensor methods such as biolayer interferometry or surface plasmon resonance. The non-specific techniques don't use antibodies to detect the HCP. We can find techniques such as electrophoresis, chromatography, or mass spectrometry. All these techniques have their own advantages and disadvantages, as well as their own limit of detection. Among the methods of HCP analysis, anti-HCP ELISA is considered as the gold standard. This is mainly due to its high sensitivity and high throughput. However, unlike the conventional ELISA, the anti-HCP ELISA doesn't detect a single target, but should detect a complex group of proteins. So the HCP ELISA generally uses polyclonal antibodies produced against CHO cell lysate for both capture and detection antibodies. These antibodies are produced by immunization of an animal like rabbit, goat, or chicken with an HCP mixture. This raises an important question. Can anti-CHO HCP antibodies detect all the HCP that are potentially present in the biotherapeutics product? During the anti-HCP antibodies production, some HCP can be non-immunogenic in the animals, and therefore no antibodies will be directed against this protein. However, a lack of immunogenicity in animals doesn't necessarily mean that the HCP will not elicit any immune response in humans. For this reason, the evaluation of the coverage is required to prove that the ELISA is suitable to detect HCP in the biotherapeutics. The coverage is the percentage of HCP proteins that can be detected by the polyclonal antibodies, and this is determined using 2D differential gel electrophoresis, or 2D-DIGE. 2D-DIGE was introduced in the late 90s and is based on the fluorescent labeling of samples and their subsequent separation on 2D gels. HCP proteins are first labeled with site 3 before 2D separation. The first dimension is based on the isoelectric point of the protein, and the second dimension is based on its molecular weight using SDS page. A Western blot is then performed by transferring the proteins to a membrane. Afterwards, anti-HCP antibodies are incubated with the membrane, and a secondary antibody labeled with Cy5 is used for the detection. Using the specific wavelengths of excitation and emission of each dye, it is therefore possible to measure the total number of spots on total proteins image using site 3 labeling, the number of spots recognized by the anti-HCP antibodies using Cy5 labeling, and the number of matched spots which are common to site 3 and Cy5 images. The coverage can be therefore calculated using this information. However, the main difficulty is to select properly which spot can be considered as labeled or not. In this project, Melanie Coverage software was evaluated for coverage determination. Melanie Coverage software was developed by Citiva in order to guide the analyst in the spot detection decision and make analysis less subjective. This software processes the images with a user-friendly step-by-step workflow. This workflow includes, first, the quality control of the image. Images are imported in the software, which checks the quality of the scans in terms of saturation, background, and level of gray. If needed, images can be cropped and the contrast adjusted. The second step is the alignment. As the 2D-DIGE images are from the same blot, no alignment is necessary. Next, the spot detection. Spots are detected based on the specific parameters, including the smooth parameters, the saliency parameters, and the minimum area. These parameters are automatically determined by the software, but it is possible to manually adapt these parameters. Then the spots edition. Even though the detection of spots is based on automatic parameters, it is still possible to include missing spots or exclude artifacts. The fifth step is the presence filters. For each image, a 3-level spot filter is applied to categorize spots as absent, uncertain, or present. This is based on the intensity of the spot. The last step is the coverage calculation. Uncertain spots will require manual review. In order to make the analysis easier, it is preferable to work on the 3D image of the blot and to navigate from one spot to another using the spot table by increasing and decreasing the contrast of the 3D image. After the confirmation of uncertain spots, percentage coverage and Venn diagrams are created and exported to the final report. During this project, we have tested 2 different anti-HCP antibodies. The first one with a theoretical high coverage and another with a lower coverage. HCP proteins were first labeled with site 3 or with Cy5 and were run in separate gels. About the detection, antibodies were either labeled with site 3 or with Cy5 depending on the HCP labeling. We swapped the labeling for both HCP proteins and antibodies in order to determine if the variability comes from the staining. Membranes were scanned and images were finally processed by 2 different analysts using Melanie Coverage software. An example of pictures is presented at the left side of the slide. In this picture, we can observe the total proteins stained with site 3 dye, the proteins detected with an anti-HCP antibody coupled with Cy5, and the overlay between the two pictures. An example of Venn diagram is also presented in the middle of the slide. If we move to the results, we can observe that coverage is quite consistent between the two analyses, between 20 and 30% of coverage for the low coverage antibody, and around 80% for the high coverage antibody. The variability between the two analyses is very low and comes from the number of uncertain spots. A small difference comes also from the dye used to detect the proteins. When antibody is labeled with Cy5 dye, the coverage is systematically higher than with the Cy3 dye. In conclusion, 2D-DIGE is a valuable tool to evaluate the coverage of the anti-HCP antibodies. This evaluation is necessary to prove that the ELISA method, which will be used for routine testing, is appropriate. In this context, Melanie Coverage software can guide the analyst through the image analysis process with a user-friendly step-by-step workflow, delivering quick and reliable results with a reduced subjectivity. Finally, if you need more information on 2D-DIGE and Melanie Coverage software, an application note is available and can be downloaded on our website.